is bio 1. All right. So, let me hide that. Cell communications day 2. Yesterday, we talked about the 3 steps of cell signaling, which were reception, transduction, and response. So today is going to be reception, tomorrow transduction, Friday response. Today's Wednesday, right? Yeah. Okay, cool. So today is cell communication day 2, reception. It is pretty picture heavy, so I'm gonna keep it to the smartboard just to make sure that the actual pictures are recorded, because we've got a couple different signaling molecules and systems to draw out. So if you have multiple colored pens, that would probably be for the best. If you want colored pencils, I can knock those out really quickly, just in case. So, the process of reception is when a signaling molecule binds to a receptor and the receptor changes shape. So, a signaling molecule binds to a receptor protein and the receptor changes shape. That is the process that we are going to talk about today. Getting from signaling molecule binds to receptor, receptor changes shape, and signals the next step, which is transduction. So the reception of a signaling molecule depends on the receiver. So, reception of signaling molecules also down depends on the receiver. We talked about how in yeast cells we've got 2 mating types, alpha and A, and the alpha cells release a signal that only the A cells have receptors for, and the A cells release a signal that only the alpha cells have receptors for. So unless a cell has a receptor to receive this signaling molecule, nothing happens. The receptor is responsible for the continuation of the signaling process. Receptor molecules are super, super specific, because they have to have active sites that correspond directly to the signaling molecule, and they have to have the machinery to change shape and signal the next step. Yeah, exactly. So signaling molecules are complementary in shape to a specific site on the receptor. So they fit together just like puzzle pieces. Signaling molecule has a complementary shape to a specific site on the receptor. So, and that's pretty much true of all signaling molecules, no matter where they are or what kind of cell they exist in. They all have sites where the signaling molecules bind. or interact, and they are all relatively specific. So, a little bit of jargon because scientists like to name things a million bajillion times. A signaling molecule will act as a ligand. Some professors will say ligand. I do not like that pronunciation. So I say ligand because that's how I learned it. But if you hear ligand, it's the same stuff. So signaling molecules act as ligands. A ligand is any molecule that specifically binds to another molecule that is often larger than it, that causes the receptor to change shape. So some of these aren't necessarily signaling. So not all ligands are signaling, but all signaling is because of ligands. Ligands are molecules that bind to another, often larger molecule. Causing it to change shape. So a lot of times people shorten signaling molecule to ligand, but they mean the exact same thing. All right, so signaling or receptor molecules, signal receptors can be found in multiple places. They can be found in the plasma membrane, embedded in the membrane, or they can be found free floating within the cytosol of the cell. So we're going to talk about both, but we're going to specifically focus on 3 different types of receptors in plasma membranes. That's where the bulk of what today is. So we're going to start with receptors in plasma membranes. I'm going to make this little stand out. Receptors in plasma membranes. No point in that. So, receptors in plasma membranes, there are 3 major types. So, the first one we're going to talk about is the most common. I'm talking like there's 800 plus different types of these types of receptors. And because they are so common and because they pop up a lot, they're the ones that I'm going to be going into in the most detail. They are called G protein coupled receptors. Or GPCRs. Yeah, this is the largest family of receptors. So I'm going to go through the process that happens when a G protein coupled receptor works. There are 1, 2, 3, 4 different steps. Would you rather me draw the picture and then write the word explanation, or write the word explanation and then draw the picture? Words first? Cool. All right. So, step 1, I'm going to use Roman numerals because we're already numbering. In step 1, we have a G protein that can move along the cellular side of a membrane. So there's G protein coupled receptors, there's G proteins, and there's an enzyme. That's the players at hand here, and we'll draw those out. So the G protein can move along the cellular side of the membrane. So G proteins are embedded membrane proteins, but they don't go all the way through. They're kind of halfway through, and they can move anywhere on the membrane as long as it's attached to the cellular side or the inside of that membrane. And it can be active or inactive and that all depends on whether or not it is bound to a GTP or GDP molecule. GTP, GDP are the same, are very, very similar to ATP and ADP, but instead of adenosine, it's guanine. So instead of adenosine triphosphate, it's guanine triphosphate. So it can be active or inactive depending on GDP or GTP. So we're going to set the stage. We have our cell membrane. Yeah, all right. I'll get it one of these days. We have our cell membrane. This is outside of the cell. This is inside the cell. I'm going to use the big chunky marker so I can switch colors easily. We're going to draw this structure right here. This is going to be our G protein coupled receptor. So this is our receptor molecule. In this form, it is inactive. Okay. Continuing that membrane on a bit, we're going to have something that spans halfway through the membrane. And this is going to be our G protein. So I'm just going to write G and then my little protein symbol. You can write G protein. Attached to that G protein, make it orange. Attached to that G protein, we are going to have our GDP molecule. GDP. My board's not calibrated. This is also inactive. So this is before anything gets started. So we have our receptor, we have our G protein, and finally we have this is our enzyme. It is also inactive. So this is when there's no signaling molecules present, nothing's happening, everything's inactive, everything's shut off. These are our key players. We have our receptor, we have our G protein, we have our enzyme. That's step one, essentially. Okay. Good here? All right, so step 2. Still sticking with the Roman numerals. Our signaling molecule will bind on the outside of the cell. Signaling molecule binds outside the cell. This will change the shape of the receptor. So, signaling molecule binds, changes shape of the receptor. This change allows an inactive G protein to bind. When the G protein binds to the receptor, the GDP that was in the G protein gets displaced, and a GTP takes its place. So, GDP displaced, GTP binds. So when the G protein, the inactive G protein, is able to connect to the receptor, it will bind to the receptor, the GDP will get knocked off of it, and a GTP will take its place. We have not touched the enzyme yet. So, we still have our same orientation. This is outside, this is inside. Make sure I got the same color scheme. Blue, yes. All right. So, our signaling molecule will bind. We'll make it, I don't know what color I haven't used. We'll make it yellow. Our signaling molecule will bind to our receptor. That's really thick. So, when our signaling molecule binds, we have a change in our receptor shape. Now, this is an activated receptor. Now that that receptor is activated, our G protein will bind. When that happens, our GDP will go out of the cell, or of the G protein, sorry, and a GTP, which we are going to have little squigglies around, will come in. So, we're not adding a phosphate group. We are kicking out a GDP and adding a GTP that already exists. Make sure I got my colors still right. Enzymes red? Yes. Okay. Our enzyme is still inactive. Nothing happening yet. Good so far. So our signaling molecule binds, which activates our receptor. When our receptor is active, it allows this G protein to bind to it because it opens up an active site when they change the shape. And once that G protein is in there, it will knock out that GDP and accept a GTP. So that's step 2. In step 3, our G protein is now activated. Hold on. Yes. That activated G protein will bind with the enzyme, changes the enzyme shape, and triggers the next step in the cellular response. So that G protein that can still move along the inside of the membrane, now that it is activated, can leave the G protein and go to the enzyme. So activated G protein binds to enzyme, which changes shape, and triggers the next step in a cellular response, which we know from yesterday to be transduction. Our signaling molecule from our G protein coupled receptor, so our blue one, is reversible. So our yellow signaling molecule will just leave. Once it's done its job, it'll dissociate. We'll be fine. The fact that this whole process is reversible is really important. It means our cell can reuse this machinery over and over again. It doesn't have to make new enzymes every time one is triggered by a signaling molecule. So anything that happens is completely reversible. All reusable. So we'll get our picture. Start with our same inside outside. Our G protein coupled receptor is back to normal because our signaling molecule has left. That's gone. We don't need it anymore. So this is where our enzyme comes into play. Okay. Yeah. Draw it out. It's a little funky, but it'll be all right. Our activated G protein, oops, My size distribution's a little off, but it'll be all right. Okay. Okay. Go away. So we now have an active enzyme. Because our G protein has moved from our receptor to our enzyme. This will trigger a cellular response. So our G protein had activated, our G protein coupled receptor, was activated, allowed an inactive G protein to bind. That G protein kicked out a GDP, brought in a GTP, and became activated. Now that G protein was activated, it left our receptor and went over to the enzyme. Once the active G protein left, so did the signaling molecule. So now we have our inactive receptor ready to start over. Our activated G protein binded to that enzyme. That enzyme changed shape because that's what happens when something binds to an active site. That change in shape triggers a cellular response. Cool? Awesome. All right. Last step. Because we've got our cellular response, all we have to do is get back to normal. So, step 4, our enzyme will hydrolyze GTP into GDP. In order to trigger that cellular response, it needed some energy, and that energy comes from our GTP. So, enzyme hydrolyzes GTP to GDP. Enzyme hydrolyzes GTP to GDP. And then we have our extra phosphate group. And our G protein will leave. So if we're drawing that out, again, same orientation. So that will go that way, and we will get a phosphate group. In this image, everything is inactive. It's essentially the same picture that we drew in the first step. It is exactly the same picture we drew in the first step, except we've got that new addition of that phosphate group coming off of it. that is how G protein coupled receptors take a signal from the outside and turn it into a cellular response or trigger a cellular response. This is how a lot of signaling pathways work. Like I said, there are over 800 plus G protein coupled receptors out there. Which is quite a bit. Okay. Anybody have any questions? Okay. All right. So I'm just going to skip pages so I know where to go. The next type of receptor we're going to talk about is a lot shorter. They are receptor tyrosine kinases. Receptor tyrosine kinases, or RTKs. They are kinases. Kinase ends in ase, which means they have enzymatic activity. A kinase, so just in general kinases, a kinase is a protein that catalyzes the transfer of phosphate groups from ATP to another protein. Kinases catalyze transfer of phosphate groups from ATP to other proteins. Yeah. So, a tyrosine kinase is an enzyme that takes phosphate groups from ATP and adds them to the protein tyrosine, or the molecule tyrosine. So, a receptor tyrosine kinase is a kinase that takes phosphate groups from ATP, tacks them onto tyrosine, and acts as a receptor. So this one I don't have words for. We're just going to draw it because it's relatively self-explanatory, not as complicated as our G protein was. So we are going to have, I got to use small little things, two different inactive RTK monomers. So we're going to have this little receptor thing, a little stem, these three, and then I'll come up like that. Not the cleanest in the world, but it'll do. So these are our active sites, and these are our tyrosines. We've got three right here. And our cell membrane acts right there. Right there. I'm going to make this smaller eventually. But we have two of those. So, like I said, not the cleanest thing in the world. I will get better once I get my bearings. So, these are inactive receptor tyrosine kinase monomers. How they are in their natural form, nothing to worry about. Try to make this a lot smaller, Nick. Okay, so then I'm just going to draw the same thing over again. Right? Our signaling molecule, make it red, signaling molecule will enter our active sites. Once our signaling molecules are bound, we will get a dimer. So our two will come together. Let's see if I can get this without messing it up. Didn't make it chunky, but essentially both of those molecules will come together and form a dimer. A dimer is just the same thing like monomer, but instead of mono, it's di, so two monomers, dimer. So now that we are, this is like a partially activated dimer. It's not completely activated because we don't have our GDPs yet, but this is our partially activated dimer. Here we go. I don't know why it's so big all the time. All right. There's our dimer. What happens? Oh my goodness. What happens? So we've got our activated dimers. So whoop, whoop. That's terrible. Activated dimers. We've got our little signaling molecules. And what happens is that we will get 6GT molecules and get 6GD molecules, and we're going to no. It's ATP. 6. I read my 6's as G's. So we're going to hydrolyze our GDP, or our ATP molecules. We're going to take those phosphates off. What we're going to do with those phosphates is actually pretty cool. So we got our same process. Try to make these the prettiest. So we've got our little dimer. We've got our signaling molecules still in there. This is the same membrane. Now, each one of these little nubs is our tyrosine. And because this is a tyrosine kinase, we are going to take our phosphate groups from our ATPs and we're going to tack them on to our tyrosines. This is a fully activated phosphorylized dimer. This took a lot longer than expected, so we're going to split this into 2 days. So, what happens, I'm just going to write out the next step in words. The activated dimer, fully activated dimer, our phosphorylized tyrosines, bind to multiple relay proteins. So we would have other proteins come up through here and bind. You could like get 2 on one side. And that will trigger multiple cellular responses. Multiple cellular responses. All right. So what I have left, I have one more of our membrane types of proteins. It is a lot shorter, and a brief stint about intracellular receptors, but that is not enough time to do it today. So we will start out tomorrow doing that, and then hopefully see where that takes us. So you'll do the exit ticket for that tomorrow after I finish this up. And hopefully we will get to transduction, but if signal transduction pathways are a little bit complicated, I will work around that. So, no worries. Thanks for doodling and drawing with me.